Hello, future biotechnologists and microbiologists. Welcome to Cosmic EduTech by Dr. Priyanka, where we discuss everything from biology to biotechnology. I'm here with very simple concept that is gram staining, but those students who have just taken or who have just entered first year of life sciences, maybe biotechnology, microbiology, or any stream of life science, this is going to be your very first staining techniques that you are going to learn. And I always found students who always get confused between the gram positive and gram negative and always ended up reporting the wrong things. So for such students, this will be a very simple way to understand the link between the theory and the practical aspects of gram staining. Gram staining was developed by Christian Gram, and this is an example of differential staining techniques. That means this method is going to help us differentiate or distinguish between the two different kinds of bacteria that are under study. So based on that, we have two types of bacteria, that is the gram positive bacteria and the gram negative bacteria. And the principle we'll see, in the principle we will see why it is gram positive and gram negative. The gram positive bacteria, they normally take up the purple coloration or the purple stain, and the gram negative, they always take up the pinkish reddish stain. Moving on and understanding the principle, I always recommend understanding the reagents that are used, especially in case of gram staining. So here is a list of reagents that are used for gram staining. There are four things to be remembered here. Primary stain, that's crystal violet. Modern, that is grams iodine. A decolorizer, in most of the cases we use alcohol, but certain labs they use acetone too. And last one is the counter stain, and that's saffron. The crystal violet, that is going to be your primary stain. And what is a modern? Modern forms a complex with the primary stain. And then the next step, that is decolorization, that is the deciding factor whether this complex between crystal violet and grams iodine has to be washed off or to be retained. That is where the distinction between gram positive and gram negative comes. So now let's move on to the principle, because now we have already understood what are the chemicals or the reagents used. So understanding the principle will be even more easier. Now the whole thing is based on the difference in the cell wall composition. So let's move on to the previous slide. If you see this image here, again it is I have taken from the internet, it is not my own image. The gram positive bacteria, they have this peptidoglycan, okay, which is thicker as compared to the gram negative bacteria. Peptidoglycan in gram negative bacteria. In gram positive, it is almost around 50 to 90% of the cell wall. It is made up of peptidoglycan, whereas in case of gram negative bacteria, it amounts to just around 10% of the cell wall. Now, as I had discussed the reagents, okay, the primary stain, that is crystal violet, it binds to the peptidoglycan layer in both the types of bacteria. But see the difference and the thickness. Based on that, you can imagine that obviously the amount of crystal violet taken up will be more in case of gram positive as compared to gram negative bacteria. So next step is the formation of the complex between the crystal violet, that is primary stain, and the modern, that is iodine, grams iodine. Now this complex form, okay, the formation of the complex between the primary stain and the modern, that's your crystal violet and grams iodine. So it is followed by the next step, that is decolorization. So in decolorization, what happens is the decolorizer that we use here is the alcohol or acetone. It forms or it dehydrates this peptidoglycan, and the complex form, that is the modern complex between crystal violet and grams iodine, it is trapped within the peptidoglycan, and when we are adding the decolorizer, not allow this complex to be washed off from the cell wall. It gets trapped, it gets retained over there. But in case of gram negative bacteria, peptidoglycan layer is very thin, the complex between the crystal violet and Grimes iodine, it is being washed off and it is again available for the next stain to be taken up. Stain is easily taken up by a gram-negative bacteria as compared to your gram-positive bacteria. So the whole thing, the difference of the appearance, that is either pink or purple, is again linked to the thickness of peptidoglycan layer. So because it is thick in case of gram-positive bacteria, it is going to take up the complex, or it is going to retain the complex between crystal violet and Grimes iodine. Hence, it is going to be seen as purple cells under microscope. Whereas in case of gram-negative bacteria, the because the thickness of the peptidoglycan is very less, this complex is being washed off and counter stain has been taken up. That's what it is making it pink in appearance. So this is the basic difference and the common link between the theory and the practical of staining. So now let us try to understand the staining principle. The staining principle or the staining procedure is very simple because now we have already understood the theory. Again, it adds more, it makes the procedure, understanding the procedure even more simpler. So first thing is we have to always use a fresh culture. Take a grease-free slide. Okay, always make this a habit. These are some of the good habits a microbiologist or a life science student should have. Take a clean, grease-free slide. Make a habit of cleaning it with alcohol and make a nice smear with the nichrome loop. Make a nice smear which is thin and evenly distributed. Allow it to air dry and heat fix it. Once this is done, flood it with crystal violet. Allow it to be there for 1 minute so that the peptidoglycan should take up this crystal violet. Once this is done, we are going to add the Grimes iodine, that's your mordant. But before this, make sure you are washing off that excess of crystal violet very gently. So after that, we have to add the mordant, that is Grimes iodine. Allow it to be there for 1 minute. And during this time, it is going to allow the formation of the complex between the crystal violet and Grimes iodine. Once this is done, again gently, very carefully wash it off and then add the decolorizer. When we are adding the decolorizer, you would see that there will be oozing out of the crystal violet. Okay, the excess of crystal violet will be oozed out because we are adding the decolorizer, which is alcohol or acetone. Once this step is done, then we are going to add the counter stain, that is going to be your safranin. Keep it for 1 minute. Now during this 1 minute, if it is gram-negative bacteria, the decolorizer must have already washed off the complex between crystal violet and Grimes iodine. So safranin is going to be easily taken up by the gram-negative bacteria. And if the culture was gram-positive, then there is no chance or there is no space for safranin to enter inside the cell wall. So this is a basic simple procedure of gram staining. So important thing you have to remember is the time duration. It is 1 minute. So decolorizer has to be kept for around 10 to 20 seconds. We have already discussed this. And what is the result? Once the 1 minute of safranin is done, we are going to again very carefully wash off the excess of safranin, then blow dry it and observe it under oil immersion microscope. So you have to look for a very neat field where you will find very even kind of smear. If it's a gram-negative culture, you will find all pink, pinkish reddish cells. And if it is a gram-positive culture, you will find all purplish cells. But if you are looking for some sort of contamination, then you will come to know there is mixture. Both type of cells will be present. Pink gram-negative and gram-positive. If you have not mixed, then you know there is something wrong with your culture. Let me give you one simple hack to remember. Pink is gram-negative and gram-positive is purple. So common example of gram-negative bacteria is E. coli and gram-positive bacteria is Staph aureus. You must have always seen teachers always use red colored pens, right? So remember the cross mark, negative, indicating negative. It will always be in a red colored pen. So red colored cross, remember it's gram negative. So gram negative cells will be seen as pinkish reddish cells. What is remaining is positive. So correct answer, that is blue or purple. So let me give you some quick tips which you should follow and certain things that you should avoid when you are performing a gram staining procedure. So very important thing is using fresh cultures because there is a chance of contamination also. So always make sure you are using fresh cultures. Now next two steps are very important. Excess of anything is always bad, right? So you should do it in moderation. So never over decolorize, also never under decolorize. So you have to be very perfect with the timings and how you perform the decolorization step, because if you do anything of this, that is over decolorization or under decolorization, it is going to give you false results, either false negatives or false positive. And most of the time, this is a common mistake most of the students do during the practical exams because they are in hurry and they end up reporting the wrong things. And usually that's a common discussion after the practical exam. Hey, I saw red colored cells. Somebody will say, hey, no, I saw purple colored cells. So that's the confusion. That is because there is either over decolorization or under decolorization. Next is making thick smears, a big no for microbiologists. You should never make thick smears. Smears should be very thin and evenly distributed. So always show the field which is very clean. So when you are calling the examiner, make sure you have spotted or you have found out the correct and very neat field. Next, what people do is, again, the students do this during the practical exam because they are in hurry. They are not going to hit fix the smear properly. And next step is excess washing. First of all, they will not hit fix the smear properly, and in hurry they are going to wash it. But in turn, what happens is the entire smear is only being washed off. So there is nothing on the slide, so you cannot perform your stainings. And I'm quite sure that nobody is going to make such mistakes again during their practical exams. And also, whatever explanation I have given you, it is going to definitely help you to answer your Viva questions. Let the examiners ask them the way they want. So, all the best for your practical exams. So, like, share, and comment. So, do comment what is the next experiment that you want to make even more simpler. So, please follow the channel. Until then, keep smiling, keep studying. All the best.